we introduce the exon 2 to 8. And then we have our full gene of factor 9 here. And then after the 6 to 8 weeks, this mouse recover the factor 9 and the coagulation time is shortened. So we could treat the hemophilia by a single intravenous injection. And the second target is XKIT. And XKIT is immunodeficiency disease, and which is caused of the mutation or lack of this gene of the interleukin-2 receptor gamma subunit. And we made a skip pig, and we extract the blood stem cells from the bone marrow, and we will perform the genome editing. And we transplant this cell to the disease skip pig to treat this XKIT. And in a pig, the skip pig lacks the NK cell and the T cell, so which causes immunodeficiency. So we collaborated with Professor Hanazono in the GC Medical University, and we made a skip pig. We succeeded in the breeding of a skip pig. We have interleukin-2 receptor gamma knockout male. We extract the cell and mix with the embryo of the female pig to make by the micro injection to make a chimera embryo, and we grow this embryo to make a skip chimera. And this skip chimera male, we breed between this skip chimera male and the wild type female. Then we have a hetero female, and we breed hetero female and wild type male. We'll have a skip male in 25%. And we could grow this male skip pig in 3 months in an aesthetic reeling condition. And we could succeed in the concentration of hematopoietic stem cell from pig bone marrow by 11-fold, which is required necessarily for the genomic editing. And we first checked the genome editing in a pig, and we first knock out of ITGB2 gene, because if we knock out this gene, we can have this knockout cell in the capillary blood, and we can check the efficiency of the genome editing in the pig. And we are now waiting for the result within this October. And for the skip pig, we could succeed in the genome editing of stroma marrow cells. And in this case, we can knock in this interleukin-2 receptor gamma gene to the vector, using the vector to make a knock-in, and we would like to transplant this cell to the skip pig in the end of this year. So I built the venture company named Edgene, and the laboratory is located in Boston, and we are collaborating with this Boston laboratory to make the super cas9 with high fidelity and with more freedom because of avoiding of the cleavage limit by palm recognition, and with smaller size. Finally, I'd like to acknowledge to the Dr. Nishimura-san in my laboratory, which is the main person of this project. And we acknowledge to the Tsukasa Omori and Yutaka Hanazono in the Jichi Medical University. And we have a long collaboration in Fanzan in the Broad Institute of MIT. Thank you for your attention.
Now, questions are open on the floor. Due to the time limit, just one. Caltech and Institute for Basic Science of Korea. He became an assistant professor at Young University this year. He is an expert in deep sequencing and epigenetics, as well as genome sequencing. Please welcome him with a big thank you.
Thank Dr. Jack for the kind introduction, and also thank the organizers for giving me this great opportunity to present to you some of the work and to discuss application of CRISPR for medical field. So, like the training, I was told to scientists stand on the shoulder of giants and further, and as you can see, Professor Ricky is one of the giants of the field. So I hope to sort of learn from him to do some interesting research. And today, since this is a medical genomics field, I'm trying to share with you some of the work on how they apply CRISPR for medicals, but medical which is not human. So basically, I'm trying to explain to you some of the work in making transgenic animals, and also infection, bacterial gene editing. So this is a special issue from Nature, and it sort of exemplifies what CRISPR really means in biotechnology. It means that you can pretty much choose any organism to study, any organism to study. And as you've seen, like pig is one of the interesting fields that Professor Ricky suggested, and like bees and plants also being an interesting target for CRISPR. And here, what one thing we've been doing is to try to apply a newly found CRISPR called CPF1, sort of studied by Van Zhang and also by Professor Ricky in the crystal structure and function. And the interesting thing about CPF1 is that CPF1 has a vastly different structural juxtaposition compared to cas9, and of course, while further studies sort of elucidated that CPF1 also contains two nuclear domains, but there are some differences, and I'll intro the structure because I thought Professor Ricky will show its structure, but so there are some differences. For instance, the CRISPR-Cas9, as you know, utilizes a single guide RNase which originated from two different RNAs in the system of CRISPR RNA and tracer RNA. But in case of CPF1, the RNA from bacteria are single RNAs, as you see here, and it's called CRISPR RNA. As you can see from a biotechnological point of view, CRISPR RNA has large potential because it's about 145 nucleotides compared to about 100 nucleotides in SPK9s and most of the other Cas9s. And another difference is the PAM recognition. And as you can see, most of the Cas9s recognize PAM in the 3' end of the target sequence. On the other hand, CPF1 family proteins recognize TTTM or TTN sequence in the 5' end of the target sequence. And it's a double-edged sword, but usually CPF1 seems to recognize a slightly longer target sequence, about 23 nucleotides compared to 20 nucleotides in Cas9. And yet another interesting stuff is that it comes in a staggered end, which is somewhat similar to what you've seen in plutonium. You see, with the baggage 1 and stuff, you get staggered end. So there's some differences. So and what we did in last year was that we tried to use CPF1 and ask whether we can make knockout mouse. So we expressly purified CPF1 from E. coli, and we tried in vitro cleavage. And yes, we were able to cleave target sequences with recombinant proteins. And we tried to transfect human cells with this RNP, protein RNA complex, and we were able to get intel from T71 assays and T6 assays. So we moved on to apply the system to generate knockout mouse. So we chose target boxN1, or boxN1 target gene, and we selected two different CRISPR sequences. As you can see, the TPT in the fragment. And we found that when you use 2 RNA, sometimes you can get even higher mutation rate. So we decided to do that route. And first we followed the classical route of using the microinjection of RNP to mouse embryo, and we got a fairly good success rate. And then the mutation pattern was sort of expected. And as you can see, the mutation pattern is slightly different from cas9, but more or less they are deletions. But injection of RNPs or DNAs or RNAs one by one to embryos is very difficult job, and you need like manipulator, you need a skilled person to be able to do it. So we decided to go different routes. So we asked, can we do multiple embryos at a time? And at the time it was quite difficult because, as you know, mouse embryos are covered with a layer called villosida, which prevents sort of practically impossible for electroporation. So we opt for different methods of electroporation. We were able to get 50 embryos transfected at the same time, and we were able to get similar results. And to check for any adverse effect, we asked whether the electroporation can cause developmental defects. But overall, the mouse which came out after transfer was almost similar with or without the electroporation, which suggested that this electroporation RNP method could be sort of a good way. And RNP is being nice because we don't introduce nucleic acids, it's much safer, but we don't have to worry about getting it integrated into genetic DNA. So that's good. And so we wanted to check whether the mutation was specific. So we did a whole genome sequencing of the mutant mouse with a sibling. And as you would expect when you do SNP analysis, you get like, as you can see, hundreds of thousands of SNPs compared to reference genomes. So we were able to filter it out using the SNP database, and we eliminated sort of SNPs that are present in both siblings. And then we came down to a very small number of unique SNPs, and then you compare this with potential off-target size of CPF1, you end up with a single mutation in the mutant mouse, and you don't have a mutation in the wild-type mouse. And as you can see, the deletion was still there. The deletion pattern was exactly the same in the whole genome sequencing compared to our targeted sequencing. And of course, you have to look for germline transmission, and we found that the germline transmission was busy. And the allele was exactly the same as the parental. So we concluded, yes, this technique, CPF1, is an effective tool to generate knockout mouse. And so we thought maybe we can sort of extend it. And then the route we took was somehow similar. So because I think, yes, we all of us are standing on the shoulder of giants. And so mouse, sometimes it's great, usually great, but sometimes you have some problems. For instance, like we know in cancer studies, APC, BRCA1, 2, they are quite serious mutations, and it can cause breast cancer and all kinds of cancers. But if you make mouse models with the mutations, sometimes you don't get phenotype. So that's could be somehow explained because mouse metabolism is quite different from human. So then what do? We turn to something different. As you see in the first slide, the CRISPR everywhere shows pig in the middle. So we decided maybe we could try to do it in pig. So since we're going the genome editing route, we're trying to do, okay, let's apply CRISPR here, try to make some cell line and to do somatic cell nuclear transfer and to generate pig. We're sort of in early stage, but we're trying to do this step. Are we able to generate some pig cells to be applied for SNT? So we picked sort of like an important gene vacuum. And the first thing we did was double check the cell line had completely same genome sequences as reference genome as we did. And we confirmed that yes, it was the case. Then we applied CPF1 RNP to the cells. This is kidney with the fibroblasts, and we were able to get pretty efficient genome editing rates with CPF1 RNP, almost about 50%. And we're trying to move on to generate sort of knockout pig, which you can use for cancer modeling for sort of xenografts. And the next half, well, the talk will be trying to understand infections. And as I think this has been like a hot news for a couple of days in Korea. Owner of a very famous restaurant died after he was bitten by a dog. And this kind of raises our concern. Infection is deadly. Infection is dangerous, and it is dangerous. And this and the cause of death was blood poisoning or sepsis by Pseudomonas aeruginosa, which is pretty common actually. So, and in hospitals, sometimes you get additional infections just because you're in the hospital. Because hospital, you get a lot of germs, and then you usually cannot get rid of them because they're so abundant. If you treat antibiotics, they come back. So, and as you can see, in the hospital-related infections, and a lot of infections come from UTIs, urinary tract infection. When you're hospitalized, you have the catheter. And the catheter has to be kept there for days sometimes, and it causes infection. And 80% of the urinary tract infection comes from catheter-associated infection. There's about 450,000 cases only in the US, so it's like a pretty serious problem. So we sought to understand how can we deal with it. So if we cannot kill it, can we talk to them not to attack us? So when you look at the infections, so they come to the bladder, they sort of expand, they form basically colony, they form biofilms. And when they form biofilms, that becomes a problem because they sort of penetrate through the bladder and cause the kidney and cause kidney infection, which can be pretty serious. So we wanted to ask, can we prevent them from forming biofilms in the bladder? So, and we chose a model system which came from catheter. This is the pathogenic E. coli, and which can cause infection. And which is somewhat interesting. I'll talk to you a little bit why it's interesting a little bit later. So in general, the formation of biofilm depend on the interactions within the cells, on an interspecies and intraspecies interaction. And that is mediated by some of the molecules, and one of the critical molecules within the interaction called foreign sensing interaction is autoinducer 2. So autoinducer 2 is generated by enzyme or multiple enzymes, and the key enzyme is COX-S, which is like S-ribosyl homocysteine liase, which really, okay, well, it is one of the pathways. Why I mentioned the name is because it's ribosyl, so I'll tell you why about it. So we generated COX-S mutants in E. coli, and we found that it doesn't really affect the growth rate, but it effectively eliminated the transcripts. And when we did the biofilm formation assay using crystal violet, you can see the wild type versus mutant. As you can see, the formation of biofilms is like not completely eliminated, but substantially eliminated. And even the quantification and the genes that are related to biofilm formations are generally downregulated. So seeing this data, we thought, okay, maybe we'll be really able to find what's going on in here doing sequencing. So we did sequencing and we realized that it doesn't really give us the answer because you can see the yellow here is wild type, blue is a mutant. What it tells us is they have completely different gene expression profile. So we have to ask some different question. What's going on here? So and then what came to notice was that this subtype of E. coli don't have receptor for the processing molecules. So usually E. coli have a dedicated system to recognize autoinducer 2 and to turn on biofilm formation, but this strain did not have it. So, and this specific group of pathogenic E. coli didn't have it. So we asked, yes, okay, probably we'll have to look for it. Maybe it's going to give us a new target for some drugs. So, since there are a lot of differences in the metabolic pathways, we looked for receptors that this is required for transporting different kinds of sugars. So there's some of the sugars, some of the genes related sugar was turned on, turned down. As you can see, we noticed that in the virus pathway, the wild type is pretty low. So we compared the wild type versus it's pretty low. Okay, so we looked at maybe something in ribose. Okay, and then we looked for the annotated genes, and then we found out yes, there is a ribose binding gene within this genome, and it's a curative of this B gene. So, okay, the curative target. So with an educated guess, we knocked out this gene in this E. coli. strain. So we did the same manner, we knocked out, and then realized that yes, we were able to find some of the biofilament genes are not repressed in the mutant strain. So that's what I think here. And then the next step we're trying to go from here is to via generator generate inhibitors to basically flood the system and then to prevent the AI2 from intake of this pathogens and trying to study the interspecies interaction of E. coli. Because when you look at catheters, you don't only see E. coli, you see pseudomonas, you see staph, different stuff. You're trying to see whether we can dissect the interaction of these species and to understand interaction. And I think this is somewhat interesting because infection, we should not think about infections and CRISPR, but yes, it can be a very powerful tool. And as in the medical field, yes, I hope we can find opportunities to sort of to connect it with different kinds of diseases, which is in conjunction with bacteria. So, and lastly, I would just want to tell you that this is like a sort of this is a difficult graph. But as you can see, in the wild type, in the growth, in the log phase, it secretes AI2, and then when it reaches the station phase, it sort of brings AI2 back in. So you see up and down of the AI2, as you can see in red, as you can see in gray, where you can see when the receptor, purity receptor is mutated, you see the AI2 remains in the medium, suggesting that yes, the dynamics of the AI2 and the biofilm formation is disrupted, bringing an effect in the AI2 uptake. So with that, I'd like to thank our group and collaborators, and especially and the big work is done by labs, and
Seoul National University. He's a specialist in ophthalmology, and he has worked as a professor at the Seoul National University since 2007. Recently, he performed outstanding works such as in vivo genome editing using CRISPR-Cas system. Please welcome him with a big applause.
Thank you for kind introduction. I am Jung Kim. I'm working in Seoul National University Hospital. Actually, I'm an ophthalmologist, I'm a clinician. And as a clinician, I'm a specialist in the congenital eye disease, LCA and FVR, and retinopathy of prematurity, and retinoblastoma, and congenital cataract. And as a clinician scientist, I'm also running a small lab. My lab is focusing on the angiogenesis related blindness. Because in the field of the ophthalmology, the major cause of blindness is related to the angiogenesis. Today, I will talk about some my result of the in vivo genome editing. As you know that recently, the clinical trial using the CRISPR-Cas was open for the lung cancer in China. Actually, that this year in China, that CRISPR-Cas, the clinical trial is already performed. In this study, that T cells are collected from the patient, and these T cells are ex vivo genome edited and infused in the patient. This kind of the ex vivo genome editing is already well studied by the genome editing researchers. And then you know that T cells and hematopoietic stem cells, iPS cells are already used for the ex vivo genome editing. But relatively, the in vivo genome editing is more challenging. Many researchers suggest that the brain, liver, and muscles could be a good candidate for the in vivo genome editing. And I think that the eye is another good candidate for the in vivo genome editing. The eye is very small organ. Did you know that we have two eyes? Although the eye size is very small, but it is very critical organ because the vision is this is the beginning point of the visions. And interestingly, the eye have a very well-structured CNS neuron. This is the retina. And this retina has about that 1 neurons and 3 neurons and 2 neurons. Totally 6 CNS neurons are located in this retina and is well arranged in this area. So that every day, the eye doctors can do surgery on this specific layer so that you can easily access to this specific neuron from the outside. So that I think that the retina is very good movement for the in vivo approach. When you think of that genome editing, the many researchers think that the genome editing could be applied to the monogenic inherited disease. It's right. But this kind of the monogenic inherited disease are very rare. You know, as I told you, that I'm a specialist in this kind of the monogenic inherited disease. In Korea, only 2 eye doctors is a specialist in this field. Me and my senior professor. But unfortunately, my senior professor will retire 2 years later. So that from the 2 years later, I will be the only eye doctor in Korea to carry this kind of inherited eye disease. I think that the eye doctors every day need more common non-genetic retinal disease, so that we should expand our genome editing techniques more common eye disease. Then I will introduce you to the more common eye disease. In the field of ophthalmology, the major causes of the blindness are, as I told you, the angiogenesis, related to angiogenesis. In children, our retinopathic prematurity, the RP, is the major cause of blindness. In the diabetic retinopathy is common in the 40 to 50 years. And over 60 years, the AMD is the major cause of blindness. And retinoblastoma is the cancer in the eye. And although the incidence is very low, in every year in Korea about 30 new patients are enrolled in Korea, and about 90% of the patients come to my clinic. So that actually I'm in the care of the retinoblastoma patient in Korea. Anyway, then this retinopathy has the common pathology is the angiogenesis. In retinopathic prematurity and diabetic retinopathy, the angiogenesis is occurs in the retina. In age-related macular degeneration (AMD), the angiogenesis occurs in the outer retina. Although the location is different, basically the basic mechanism is the same. It's angiogenesis. The angiogenesis is occurs by the variable processes. Then, did you know that VEGF, vascular growth factor, is the major growth factor to induce the angiogenesis? Usually the VEGF was studied in the cancer biologist. Did you know that the VEGF can induce usually VEGF is secreted from the cancer cell, and this VEGF induces angiogenesis. But in the cancer, variable growth factors are secreted, EGF and FGF and VEGF. All these growth factors can induce the angiogenesis. So that in the cancer, the suppression of the VEGF is not enough. Because if you suppress the VEGF, then the anti-angiogenic effect is very transient. Soon, the new vessels are growth because of the other growth factor, such as the PDGF and FGF, they induce that angiogenesis. But in the retina, the VEGF is the only major growth factor. So that in the eye, the suppression of the VEGF is a very good strategy to control the angiogenesis. But actually, some global pharmaceutical companies already developed good VEGF inhibitors. As you know, the Lucentis is the blockbuster in the eye market. And newly, the Eylea is joined in the But unfortunately, then maybe with that indicated patient of this VEGF inhibitor, this VEGF inhibitor is very easily injected into the vitreous cavity. It is so easy procedure. And this effect is so great. As you see, the edema in the retina is effectively reduced just by one injection. This effect can be seen only after 1 day after injection. It's really good drugs, VEGF inhibitor. But unfortunately, when we injected the VEGF inhibitor, when first injection, about 60 to 70 patients showed a good effect. But during the follow-up, about half of the patients suffering for this treatment. So that actually, around the 30 or 40 patients can be sustained by these VEGF inhibitors. So we need another treatment options and another target. So I do the angiogenesis related research. This is the platform in my lab. The last 10 years, I established this platform. Actually, I am doing that active collaboration with good vascular biologists. Collaborators if they ask to me some candidate for the angiogenesis, so we can start it from the cell. Sometimes we starting from the microfluidic systems, and we use that gel-efficient light for the screening the drug candidates. When you have a good candidate, we doing that mouse model. In this mouse model, we check that we validated the efficacy of the drug candidate. It has a good efficacy. Then we move to the large animal models. Then usually that I use that bigger, but recently I'm trying to change that the pig because that many animal protection association groups are strongly don't want to use the dog, so that I changed the pig. Anyway, that sometimes I use the monkey because that recently we also established the new monkey system is marmoset. So that now in my lab I use the cynomolgus monkey and the marmoset monkeys. And also that I'm doing that the PK study using the rabbit. The rabbit, the eye of the rabbit is the size is very similar to the human. So that we use the rabbit. Anyway, that using this platform, I am doing the variable that anti-angiogenic drug candidate experiment. This is the microfluidic system. This system is established by collaboration with Professor Nurijan in the Seoul National University. And our microfluidic kit is the size is that around that 500 Korean won. It's very small. And in this microfluidic kit, I can make that vessels just like the real vessel. It has the 3D structures. And you know that usually we doing that angiogenesis, or say we just check that the 2D tube formations. But in this microfluidic system, you can make that 3D vessels, and through this the lumen is that some nanoparticles can pass through this lumen. And then it takes around 7 days to make the complete vessels. And recently we also established that the new microfluidic system. In this system is mimicking of the age-related macular degenerations. And when we get some good candidate for the anti-angiogenesis, we injected the candidate variable rate that the topical and intrastromal, and intracameral, intravitreous, and subretinal. And in tightening of the retina, we usually inject the intravitreous injection or subretinal injection. But I would like to focus on that recently we established a new drug delivery system. Is that microneedle. Our microneedle is just to pass through that partially the sclera, and this drug candidate is passively passed through that into the retina. This kind of the microneedle is very safe because it clinically now we use that just the total penetration of the square. This kind of the total penetration, complete penetration, it can make some infection and the side effect. But if we do not have the complete penetration, if we do just partial injection, it will be more safer than the conventional injection. So that this microfluidic system is established by collaboration with Professor Liu in Yonsei University. Anyway, that I'm doing that variable drug candidate, I will show that some example of my experiment. This is an example of small molecules. This small molecule is supplied by the Professor Young Seo in Seoul National University. And now we have the post generation of the small molecule derivative. Now this drug is pending on the licensing out to some company. Anyway, that this small molecule is show that very good effect. This small molecule is not targeting on the VEGF. Instead, this small molecule targeting on the HIF1 alpha. And you know that the HIF1 alpha is the transcription factor of VEGF. And this is an example of antibody. In this antibody is not targeting on the VEGF. Instead, we developed the antibody targeting on the C5. C5 is the complement of the system. And this antibody is actually the licensing out to the Celsius now. Anyway, that this C5 antibody showed that very good effect, just like the VEGF antibody, and it showed that less toxicity than the VEGF antibody. And this is an example of peptide. This peptide is collaborated by the Professor John in KAIST. And basically, this peptide is the target of VEGF. But different from the VEGF antibody, this peptide side is very small. So that when you injected this peptide, it is easily removed from the eye and easily extracted from the body. Because the anti-VEGF antibody is circulated in our body for a long time, it can make some side effect. Instead, this peptide is easily secreted from the body. And we also use the protein drugs. Many researchers think that protein drugs so far never succeeded in the clinic. But this protein drug is not the cross-spectral. This protein drug is inhibited endocellular and extracellular matrix interaction. Our targeting of the extracellular matrix fibrinoactive. And basically, this protein drug is developed in the Mogam Institute. But unfortunately, this Mogam Institute forgive to go on the clinical trial. So that I'm so sorry that, because in my experience, this protein drug showed a very good effect and less toxicity. And I also used the polymer. Usually I use the polymer that for the delivery something. But for example, we attach the VEGF aptamer. It is already used in our clinic, and the VEGF aptamer in the polymer, and this is packing like this. And we injected it into the eye. The polymer is slowly released, so that we can have the sustained release in the eye. And our polymer study is supported by Professor Won Jong Kim in Hostek. He also is a member of the DIBS. And we also use the nanoparticle. I'm more interested in nanoparticle-induced cell assembly of the proteins. When we inject the nanoparticle into the vitreous cavity, and some of the natural proteins are obligated surrounding the nanoparticle, this is called the corona. When we manipulated this corona protein, we can make that new drug candidate. Anyway, we succeeded to make a new protein assembly. We call it the Sarban, and then this kind of the nanoparticle-induced cell assembly of the proteins is also another way to develop the drug candidate. Anyway, that using my platform, I have tried the variable experiment, the variable target and variable platforms. Then the CRISPR system is also one of my options in my lab. Today I will show that some examples of my in vivo genome editing based on the published data because I have some computational issue, so I just showed that the published data. Actually, in my lab, I'm doing that therapeutic in vivo genome editing experiment for the variable target genes, for the variable target cells. Especially, I'm interested in the RT cells and macrophage and neural cells and endocellular cells. And we also tried variable delivery route, the intravitreal injection and subarachnoid injections. And we used variable materials. But today, I will just show the two examples of my experiment. One is the red, one is the blue. The first example is that in this study, we used the laser-induced CMV mouse model. This is the official animal model of the age-related macular degenerations. In this study, we injected cas9 RNP targeting VEGF-A into the subretinal space between the RP and photoreceptor cells. At first, we chose that the RNP, the sgRNA with RNP, to show that the very high in the frequency around 80% in mouse cells. And when we doing that in this experiment in the human retinal cell, the ARP19 is that the cell line of the retinal pigmented cellular cell of human. In this cell, the indel frequency is going down around 40%. It's okay. So that we check that actually this cas9 RNP is going to the nucleus. As you see, when you treat it in the ARP19 cells, you can find that the cas9 RNP is in here. And then we check that this RNP actually go into the nucleus in the tissue, so that we injected that the cas9 RNP into the subretinal space in the normal mouse. In mouse, the cas9 RNP is located in the nucleus here. So we move to the next experiment. But we have one problem. As you see, the indel frequency is at around 20%. At first experiment, the indel frequency is around 10%. So that with this data, that we submitted our data into the nature, the many reviewers said that your data is good, but basically we cannot believe that 10% is not enough to show the impact. So we tried, but finally our result is around 20%. But many researchers said to me that 20% is not enough to show the impact. But I don't know. But actually, but our data showed the impact. Anyway, although the indel frequency is 20%, but when we injected the cas9 RNP into the subretinal space in the AMD animal models, it showed a very good effect. As you see, this is a control, and this is the treated groups. And when we're doing that experiment using the other small molecule and antibody and peptide variable platforms, then show that very similar effect. From this data, we know that at least the genome editing show that the same therapeutic effect in the AMD animal model. But when you're doing that the genome editing, the many researchers think that we should think of that the toxicity of the genome editing. it's a off-target effect. But as you see in our experiment, when we doing that in vivo genome editings, that we cannot find that serious off-target effect. And we also checked that the tissue toxicity. It's already known that Stanford groups already show that when they get completely delete of the deletion of the they do the complete deletion of that VGF in RT cell, then the cone and rod cells are abolished in the retina. But as I told you, that our index frequency is around 20%. 20% is not complete, so that it never showed that the cone cell loss in our experiment. I think that 20% is not high, but it's enough to show that the good therapeutic effect, and 20% is not never induced the toxicity in the retina. And next example is that we also use that same animal model. In this study, we used that AAV, and in this time we targeting on the VEGF and interferon alpha. And in this time we injected the AAV into the picture scalp, different way. And this is the summary of our result. In this study we used the CJ-Cas, and in this study showed that the very similar good effect, good therapeutic effect, just like the RMP. And from this study we know that the in vivo genome editing is show that the good therapeutic effect, just like the other drug candidate, and it showed that the very good safe profile. As I told you that I tried the variable molecular platform for the anti-angiogenic strategy, and I think that the CRISPR-Cas is one of my options. Small molecule and peptide protein and antibody and CRISPR-Cas. But in some point, the CRISPR-Cas is better than the other option. As you know that the antibody is very specific, it's very good. But it's one problem is of the antibodies. Antibodies just tighten on the extracellular proteins. It's never tighten on the intracellular protein. But CRISPR-Cas is easily tighten on the intracellular molecule. As you see that in our study, we tighten on the interferon alpha transcription factor. Yeah. And siRNA is also a good way to tighten on the intracellular molecule, but so far, then in my experience, siRNA is not safe. It showed that very serious toxicity in the retina. So that I think that so far, the CRISPR-Cas is, at some point, is better than the other treatment option. So that actually my lab is now doing that in both ways, in the common eye disease and monogenic inherited disease. The list we have a very good result in the LCA, that correction. Anyway, and as I told you, I'm a clinician. Every day I think of that my patient. This is actually my patient's photo. And some patients have a good vision. And some patients have a very poor vision. Depending on my choice. So that I should try to my best to help my patient have a good vision. And this is my last slide. And I have that the good members, especially that these four researchers clinician scientists, they are the specialists in ophthalmology. They are the trained in my department, and they already have the specialist of ophthalmology. But instead of the clinical work, they forgive the clinical work. Instead, they're doing the full-time research about the translational research. So that with my members, I will bring that my best to help my patient. Okay, thank you for listening.